Okay, so let's get going again. Next speaker is Lukas Reiter from Biognosis. So I guess some of you are going to go. How many people are going to go to the Biognosis seminar tomorrow? Yeah, quite a few people, maybe half or so. Great. So Lukas was a PhD student going back many years, associated with the Ebersol group, and he actually was responsible for developing many some of the core ideas and tools that you've heard about during the week, including the original mProfit and also things like Mayu. And now he's, I guess, the Chief Technology Officer. Is that right? Chief Scientific Officer. What are you? CTO of Biognosis, which I think many of you will have heard of, and I think he's going to tell us about how to make the best use of retention time spaces for DIA. Thanks, Lukas.
And thanks for the invitation to speak here at the DIA SWATH course. I'm going to quickly show you an overview of what I want to talk about today. So I have quite a bit of intro. Many things you might have heard, but it all basically aims at afterwards explaining this tiered iRT approach. So I have a short introduction in general on DIA data analysis, then an introduction to libraries in general, an introduction to the iRT concept, and then more the precision iRT concept, then the limits of this concept, and also something about what we call project-specific libraries, making use of this precision iRT concept. And also other types of libraries, and then the introduction to hybrid libraries and tiered iRT. So this kind of mixing together different data sets and building large libraries out of that. And then how we are testing this hybrid library workflow on a controlled quantitative data set, and then also in the end an application. And I want to start with this slide here. So this is the main quantitative workflow we are using in our laboratory. It's a single-shot DIA workflow, as you can see depicted here. Means normally we inject every sample only a single time, and we try to get as much quantitative information out of these samples as possible, as you can imagine. And we acquire the data normally for quantitation in DIA mode and analyze the data with our software Spectrum-Centric. And the main advantages we see with this workflow is that it's a pretty straightforward sample prep. I mean, if you would compare to other sample preps from analytical chemistry, it's probably not what you would call straightforward, but in terms of proteomics, it's a pretty straightforward sample prep. And then it's also scalable to thousands of samples. We have done such experiments also here in the Ebersol lab. Very large experiments are being run, so that's perfectly possible. And then I would also say it's quite a time-efficient workflow, meaning if you restrict your LC-MS time you have, or if you have a given amount of LC-MS time, it's quite an efficient workflow to get a good quantitation. And using this workflow, we basically published this last year. Maybe one hallmark to mention is that we were able to quantify more precursors than you can theoretically do on an HF instrument using this workflow. So you can see that it's what I mean with efficiency in terms of quantitation. Now a little bit something about data analysis. I'm sure you have heard this during the course in this week. Some words on what is called peptide-centric analysis of DIA data. So this normally starts with generating a library. Then you would go into your DIA data, generate extracted ion currents. That's why it's called targeted analysis or peptide-centric analysis. You generate these extracted ion currents. You can look at them over time. They pretty much look like MRAM data. You can look for peak groups within this data. You can integrate those peak groups. You can score them. And you can combine these scores. You can perform machine learning. And then in the end, you can perform also FDR estimation on the combination of those scores. I think there was a whole lecture about this during this course, so I don't have to go into more detail about that. And then what is called spectrum-centric analysis of DIA data, quickly shown on this slide here. So normally in this workflow, you would start on the MS1 maps, detecting MS1 features of peptides. You would somehow pre-process the MS2 spectra, like for instance Alexei is doing in DIA Umpire. You would search these MS2 spectra. You can use established search engines for that. That's a big advantage. And You can also perform FDR estimation. That's also pretty much what is established for DDA data. That's also nice. And yeah, so this in total is basically the more spectrum-centric way of analyzing the data. As you can see from the publications, it's actually the older workflow. However, the real breakthrough, I would say, was more the peptide-centric way of analyzing the data. Now, in terms of my presentation here, you can make this nice matrix, which is actually also described in this paper here from the McCoss lab. So you can distinguish your way of analyzing DIA data and actually also DDA data in this 2x2 matrix, whether it's spectrum-centric or peptide-centric, and whether you start in silico, you could say, from a FASTA file, or almost in silico, so you only have the protein sequences available, or whether you start from a library. And in my talk here, only these two ways of analyzing the data are really used. You can also do the other things, but that's only what I'm talking about today. And now a very, very short intro to libraries, because it's important what I'm talking about today. How does a library look like? I'm sure you all know by now at the end of the course. So that's an example of such a library. I only kept in columns that are the more important ones in this dummy example here. And we typically split up the information in a library according to whether it's completely necessary. Actually, only these two things for our way of analyzing data are really absolutely necessary to perform an analysis. Only the precursor MSET and the fragment MSET. And then there is important information, and I highlighted here iRT because it's in the title of my talk. So this is indexed retention time. Also, the relative fragment ion intensity is what I would call important. And then nice to have is all the meta annotation about your assays, so the peptide, the precursor charge, modifications, and so on and so forth. And you could say in very general, there are three major information contents in a library. It's which peptide precursors you look for, then the fragmentation and the retention time. And I'm talking a bit more about retention time here today in my presentation. On this picture here, you can see three similar but not exactly the same gradients. I'm sure you all know these depictions of gradients used in proteomics. So these three gradients differ with the dead time in the beginning here, and they also differ slightly with washing here, and then the slope of the gradient. And normally you would say that the peptide elutes at a certain %B in chromatography. But subtle differences in the chromatographic conditions can lead to varying retention time, also due to the dead times and so on. So the time the peptide needs from your column to the detector. And this means that RT, or retention time, is often not so easily comparable, even if you use similar but not exactly the same chromatographic conditions. And the idea we had then a few years ago was to normalize retention time to make it more useful and more comparable across systems. And this is what we called indexed retention time. And here you can just very easily see explained how this works. And it's actually much easier if you look at this picture here. So we just said, let's take one reference point or one reference peptide and say this is what on the Celsius scale would be 0 degrees. And let's take another peptide here, a reference peptide that is defined, and say this is what on the Celsius scale 100 degrees. And we just didn't call it degrees then, but we call it iRT. So we said this peptide here has iRT 0, and this peptide here has iRT 100, and everything else is just linear inter and extrapolation of this iRT. So this was the first definition of the iRT scale. And here you can see what actual peptides we used. We didn't only use 2 peptides, but we back then used 11 non-naturally occurring peptides that we actually designed. So we really made up these peptide sequences and tested their characteristics, their stability, and so on. And we also checked that they are evenly distributed on the retention time scale. And here you can see the definition. We defined actually the second peptide as 0 because the first peptide is a very early eluting one, and often, depending on the gradient settings, it's not so stable. So we decided that we only define the second one and the last one. And the applications in the beginning was actually scheduled MRM, and then, but very briefly, it became clear that, or very soon, it became clear that it can also be used for the targeted analysis of DIA data. And that's why I'm talking about it here today. So the question I ask here is, does it make sense in the peptide-centric way of analyzing DIA data to look for the peptide over the complete gradient? And you can imagine what the answer is to this question, but here you can see a full extracted ion current for one peptide on the MS2 level. I think here it's a 4-hour gradient. And then you can zoom in again, and in the end you can see your peak. And depending on which ion current you would perform your analysis, and this is what is depicted here on the right, if I would do my analysis on this level here, this would mean my analysis would be slow because I have to go through many spectra, and it's also not very specific because I'm looking at a lot of data that I could actually throw away. So it means that I increase my potential of looking at the wrong thing. Whereas when you are analyzing your data down here, it means that you have a fast analysis because you're only looking at the spectra that you need to look at, and it's also much more specific. And so this is basically what we implemented in the Spectronaut software, and it's described here in a bit more detail. So we have this 3-way of analyzing the data, which was actually initially inspired by MaxQuant because it also has this 2-way of processing the data, first the calibration and then the main analysis. What we do is a 3-step analysis. We first look for the iRT peptides, and so we can determine where the iRT peptides are, and based on this we make a secondary analysis based on the peptides in your library. We can narrow this down a little bit because of the first analysis, so we look at 1,000 of peptides from your library, we narrow it down, and then in the last analysis step, because we have really a lot of data points here, we can very accurately determine the position where we expect the peptide to elute and also the tolerances we have to give ourselves, and we can do this dynamically. Which is basically depicted down here. And yeah, so you can describe the library generation and the data analysis going back and forth between retention time and iRT. And when you generate a library, it means that you combine heterogeneous gradients, can also be homogeneous, but can also be heterogeneous, across normally many DDA runs. And now we can actually use thousands of anchor points to do this, to be more accurate. And to do this, you would basically make a local regression, iRT versus RT, and you can generate your library with what we call precision iRT, because we have a lot more than just 11 data points to do the local regression. And when you go the other way around, meaning during your DIA data analysis, you would use your library, you would perform the calibration on these thousands of peptides. You would do this with wide extracted ion currents, and then you can again do your local regression, RT versus iRT, and you can do it much more refined on small extracted ion currents and perform your final analysis with or the targeted extraction with what we call precision iRT. So you can go back and forth like that with quite high precision. And the reason why this works in Spectronaut now, also if you do not have these original 11 iRT peptides in your sample, is because we have a database in the backend that is very homogeneous, and it's just a database of peptides and their corresponding iRT values. And here's a short summary of what we all have in there. So it's a pretty good coverage of many different organisms. We also have typical contaminants and unspecific peptides in there, such that you have a high chance also in other samples to have iRT anchor points in. And this helps to make precision iRT libraries for all sorts of samples. Now you can ask yourself, where is the limits of this? That's actually a rather interesting question. Where is the limits of this precision iRT concept? And what I did here was just I plotted 2 consecutive technical replicates, and I plot the delta apex retention time versus the average retention time of these 2 runs. And this is then how this plot looks like. It's actually very similar to what has been done for much longer when you do MS1 alignment, for instance, in DDA. That's a very similar process or similar analysis done there. And what you can see is that the spread here is roughly 1 minute. Here, I think this was a 2-hour gradient, as you can see here. And that's, I would say, the lower limit of what you can expect from a nano LC in terms of accuracy you can get. You will probably not be able on a nano LC to get any more precise than 1 minute, rather a little bit above that. Yeah, then something about what we call project-specific libraries. And this is the standard protocol we are using in our company. We would first pool the samples of our experiments, and we would do this in conditions. So if you have 2 conditions, we would pool by condition 1 and pool by condition 2. Then we typically perform high pH reverse phase fractionation using these published micro pooling schemes. And then we acquire DDA runs of those fractions, and we build a project-specific library using this precision iRT concept. And then we perform the DIA data acquisition in block randomized order to avoid any bias in label-free quantitation. And the disadvantage of this workflow, of course, is that you have to generate the library over and over again. That's an overhead you would not like to do if you could avoid it. But the advantage is that the library performance is very good. It's really a high-quality library for your sample. Specifics about your sample prep are really reflected in the library. Your sample prep protocol on your samples, this can make a difference. And we can also use this iRT precision concept, or it's very powerful in this kind of setting. That's the advantage. And yeah, here you can just see this workflow as a whole depicted. That's still the standard what we are doing, even though now this also starts to change a little bit, as I will show in the end. With this workflow, we can achieve median extracted ion currents as expressed in % of the gradient that are in the range of 2%. So before I have been saying it probably cannot go below 1 minute on a 120-minute gradient. This would correspond to 1%. In reality, since you pool many DDA runs and it's not runs back-to-back on your nano LC, you can achieve 2% of your gradient length. But it's still pretty good. And then here in this publication shown down here, we looked a bit more into that in a bit more detail. So here actually we took such a precision iRT library and we injected noise into these libraries in terms of iRT. And then we looked at the performance of this project-specific library with injected noise, so meaning going down here. We also looked at having no iRT at all, meaning doing the full ion trace extraction. And we also looked at the old concept we had back then, what we called linear iRT. And the summary or the conclusion back then was that precision iRT yields roughly 25% more as compared to having no iRT at all. So that's a significant difference. And then what we were also interested in back then was which experimental factors can influence this precision iRT. And that's what we have done here quite extensively, actually. This was quite a bit of work. So we changed a lot of experimental conditions while building the libraries. And you can see all those things we changed down here. So we changed the mobile phase, we changed the gradient length, we changed the stationary phase, temperature of the column length, flow rates, and so on. And then we just ordered them by the influence on the iRT precision they had. So maybe this scale here is a bit, how do you say, a bit abstract, and it actually is. But what is really important is more the order in this direction here. And the interesting thing was that the mobile phase and the very interesting thing was that the gradient length had the biggest influence on the precision of iRT. And that was, apparently we learned later on that there have been publications showing this in analytical chemistry before. For us, it was not so intuitive that the gradient length would have a big influence on the precision of the iRT, but it actually does. So that's good to know. So if you build project-specific libraries, if you do that, if you really go the length of doing that, it's probably a good idea of doing it on the same gradient length as you do your DIA runs, because it really gives you something extra. And then, yeah, what does this actually mean if you think about that? The gradient length has an influence on the precision of the iRT, and that's what I tried to explain on this slide here. So if you would have a short gradient and peptide A elutes before peptide B, it means that on the longer gradient, peptide B suddenly elutes before peptide A. So that's, of course, in very close vicinity to each other, but it means that peptides can change their order in terms of the elution from the chromatography. And that's, of course, something you can never correct or not so easily correct with some sort of local regression. So this again reflects that that's the lower limits of the precision you can get with this concept. Then I want to show quickly another effect that's more practical, but I think it's still interesting. So many people are doing these sort of gradients. So they step to a certain B in the beginning because there are not a lot of peptides in this region. Then they do a linear gradient. Then they step again up where it's not getting interesting anymore. Then they wash, then they equilibrate. And when you're doing this, you actually have an issue at the boundaries of your gradients with respect to precision iRT. And I can very easily explain this. So here or also here, when you step your gradient, all the peptides will essentially elute at the same time, having various characteristics in terms of where they would elute in a normal gradient. And this means you cannot determine your iRT precisely anymore. It's an undefined condition. And that's why we normally recommend to do these sort of gradients. And maybe why are we recommending this? Because if you're not doing that and you build a library based on such gradients, you will end up with iRTs for your peptides that are frazzled out at the ends because it's not defined anymore. And every run will give a different line there. So if you're not doing that, if you're more using an S shape of gradient, you will not run into those issues. It's a small thing, but it can improve your libraries if you're doing it. And then where are these effects really important? They are really important at two steps. The first one is when you are building your library from heterogeneous DDA data. Then if you consider all of the things said before, you can make a better library if you better homogenize your iRT, or if you're using precision iRT for that. But of course, you will get the best libraries if it's really based on very homogeneous data. That's for sure. The more homogeneous this data is, the better your libraries will get. And then the second step where it really makes a difference is when you switch the chromatography between the library and your DIA acquisition. I mean, I'm not saying that's something you should never do, but if you can avoid it, it's a bit better. I'm not saying it's a bad idea to do it or to use a library that has been published. I'm absolutely not saying this. I'm just saying it can make a small difference if you're doing that. Then here I have a little overview of, maybe that's also interesting with respect to what I'm showing afterwards, how libraries and also our software, the methods of generating the library and the library size, how this evolved over time in our laboratory in the last 6 or 7 years. And this is not actually what we have as our standard protocol. This is more of what we have seen is possible at a specific year or at a specific time. So in the beginning, we were really only making relatively small libraries. We did repeated injections of the same samples and built project-specific libraries. If we did any tests with larger libraries, usually it didn't look very good because also back then the DIA data didn't have such a high quality as nowadays. So I would say that's the main, and also the software improved a lot in between. So I would say these are the major two reasons why back then smaller libraries performed better than large libraries. Within the course of the time, we went on to developing this high precision iRT concept. We started to make deeper libraries with fractionation in this size. This started to work really well. In the beginning, we didn't see that, but then it really started to work well. Later on, we actually started to do deeper fractionation, so really deep libraries, and we could see that suddenly this also starts to work. And we also, at the same time, developed our own way of the spectrum-centric way of analyzing DIA data, which we call DirectIA. And now in the end, so this is what I'm showing today, is this concept of tiered iRT and what we call hybrid libraries. And these are then really quite large libraries. And we can see now that these actually start to work pretty well. So there is an overhead when you make these project-specific libraries. For every experiment, you have to repeat the library generation. That's not really super great. So are there any alternatives? And I'm sure you have heard about them in this course. One alternative is of searching your DI data directly. For instance, with DI Umpire, or now can also be done with Pulsar in Spectronaut. The advantage here is that it really gives you very, very reproducible quantitation. And that's a side note. You will also have very precise iRTs because they're actually determined on the same runs as you do your experiment. So there's usually not so much variation in chromatography within your experiment. Otherwise, it would probably not be optimal. The downside is that you don't get such a big proteome coverage as you could otherwise. It also gets a lot better actually recently, but the depth is a little bit limited. Then you can also use large libraries that have been published or large data sets that have been published and build libraries based on them. The advantage here is that those are really rather comprehensive. However, comprehensive they are, but if you have a special sample prep or really special samples, it can also be that that's not really reflected in your library. And another downside is if the chromatography was very different from yours, then the iRT can be a little bit less precise. But still, nevertheless, you can ask, what is if I combine these two approaches? So what is if I take a resource library and on top of that, I just search my DIA data directly and combine the two and make a single library out of it. And we have done that actually already last year, but only with a very quick test. So without any optimization, we had, here I have one example data set. It was 3 times healthy liver tissue and 3 times cancerous liver tissue. We performed a spectrum-centric analysis of the DIA, a direct DIA analysis, and then we also took a resource library, in this case a very, very comprehensive one. It was this publication from 2014, 2,200 runs, and we built a library out of that. And here you can see the results. I only show the results here in terms of the consistency of quantification, so meaning proteins that were quantified in all 6 of the 6 runs. And what we could observe there was that direct DIA is doing a really, really good job in terms of consistent quantification. It doesn't look like that if you look at the absolute numbers. They would be more like this up here. But in terms of consistency of quantitation, it looks really good. But what surprised us then was that the analysis with the combined library was not equally as good as the direct DIA analysis, what was rather worse. So it really underperformed in terms of consistency of quantitation, and we asked ourselves why that's the case. And we could see a number of limitations with this workflow, which I have described on this slide here. So it was 3 major limitations with this quick and dirty test we did. The first one was that the precision of the iRT was diluted with the data of the resource library, because this was done on a different chromatographic system. And if we build a consensus library and a consensus iRT, it was kind of diluted for our target system. So this was suboptimal. A second problem was that it was difficult to do the protein inference and the protein FDR especially homogeneously on the whole data set. That's not so trivial. And then the third limitation was computation time, because if we had to process all the data together, this was more than 2,000 runs that had to be processed in a single go, and making the library out of that takes quite a bit of time, as we show afterwards. But this would essentially be the workflow suggested. So you take some resource data, you take your DI runs, you put them together, you build a hybrid library, and then you apply to the DI data and you get some results. And here are these challenges described in a little bit more detail. So what I have been saying before, if you analyze your DA data directly, you get really, really accurate iRTs, as you can see in this RT to iRT calibration. If you do it for resource data, so this was really the case I've been showing before. It was this draft human proteome data applied to our data looks like this. It's pretty similar chromatography, but it's not perfect. And then if we take the combined library, you can see that in terms of precision iRT, it looks like the resource data, so really not as optimal as it could be. The second challenge, as I have been saying before, is the protein inference and protein FDR. I have a little bit more details about that. So ideally, you would do this homogeneously on the 2 data sets. And yeah, here a little motivation why it's good to do that for the protein FDR, because for the protein FDR, if you start to lump together data and you don't control your protein FDR on the whole, you actually accumulate your protein FDR. And it's certainly not optimal if you do that, because you accumulate bad data. And And if you would do it too extensively, then it would certainly not lead to a very good result. And then the third one, motivated here, is the computation time. So to search the 6 TI runs, it took us roughly 0.2 days. Search these 2,200 runs took roughly 9 days. And building the whole library took roughly 9.2 days, which makes sense, just the sum of the two. But it would be a lot of work to build this library on a whole week for every experiment, right? So it's definitely not a very efficient use of computation time. And yeah, first have our solutions to the second and the third challenges we had. So what we do is we normalize the scores from different analysis. So we normalize scores from DNL analysis to the scores from the DD analysis, and this way we can do the FDR on the combined data sets, and afterwards we can also do the protein inference on the combined data set. Yeah, here it's actually, so these are not scoring histograms you would normally get from Spectrum-Centric way of analysis of data, but I just took those to illustrate what I mean. And then in terms of computation time, so what you can obviously do, right, and the other tools can also do that, you can search your resource data only once and store the intermediate results. And then you can combine it afterwards into a hybrid libraries based on the intermediate results, which we call search archives. And if you are doing this, you can bring down the generation of your hybrid library to less than a day for a normal experiment of a couple of dozens of DI runs. And it's still a true consensus in terms of library from these 2,200 runs and these 6 runs here. So it's not some intermediate result of this and this, but it's really a true consensus of all of those runs together, and it still only takes less than a day to generate the library. So that's quite neat. And then in terms of the precision iRT, which is actually the topic of my talk, so we can take these various sources that have varying iRT precision. In this case, it's the DIA data and the resource data, and we can store the iRT values of those in the library separately for each source. So we don't only store a single value in the library, but we store as many iRT values as we have sources of data. In this case, it's 2 sources, but in principle, it can also be 3 or 4 or 5 sources. And then when we perform the analysis in Spectronaut with this library, we can dynamically choose which iRT to take. So we can just take the better one in terms of the calibration. And this, of course, will lead to many peptides being done with this type of analysis and much less with this type of analysis. We can then also do the precursor FDR estimation and scoring separately for every tier and the protein FDR globally, such that the analysis is really proper. And then we tested this on a controlled data set. So we had here a data set which we compiled from a stable background consisting of mice. In this case, we chose to take distinct mice because we also wanted to have biological variability in the data set. So we wanted to have something that reflects reality quite well. So in total, these were 25 mice, 5 conditions. And then we spiked in the UPS2 standard. Not many of you are probably familiar with this standard. It's proteins in defined quantities in different abundance ranges. I show this actually here. And we chose some schema to spike in this UPS2 standard across the 5 conditions. You can also see how much in absolute attomoles on column per peptide you would get for each tier of protein. So there are 6 tiers of proteins, 8 proteins each. And we are going here in this analysis we did here from 1.5 attomole on column to 1 picomole on column, roughly. So it's a very big range of concentrations. Actually, many of those here we don't see at all in the analysis we perform here because they are super low in abundance. You would probably only get those if you would do MRM or PRM. Yeah, exactly. So here you can see the characteristics of this data set in terms of quantities in there. And then we wanted to compare how well does the analysis now work if we do a project-specific library as compared to this hybrid library approach. And this is what we did here. We built the two libraries. So we built a library from the pool of 25 samples. We did 10 high pH reverse phase fractions. It's not our standard protocol. It's actually a little bit better than our standard protocol. And we built a library out of that. And this is how the library looks like then afterwards in terms of size. And then we also built the hybrid library. So this is now really not using any overhead. We use two data sets from the Matthias Mann lab on mouse, combined the two, built a resource library, and then we did the direct DIA analysis on the DIA data, 25 runs, and we built this combined library here. You can see that in overall size, it's not really a lot bigger. That's also not what you would expect, because our sample prep is probably not so different from the sample prep here. Also, the samples are not so different. So you would not expect a huge gain in terms of coverage here. But what really makes the difference is that most of the iRTs here are really, really precise for our data. That's the biggest difference. And then we tested this. So we did the analysis as described here. What is really important, so this part is really important. We perform statistical testing. And we can do this on these 5 conditions. We always do pairwise comparisons. And since there are 5 conditions, we can do 5 times 4 divided by 2 comparisons. So in total, we can do 10 comparisons among those 5 conditions, if I calculated this correctly. And we can do this for every protein. So all the proteins identified times 10 comparisons if you can actually do the statistical test. So in the range of 50,000 or 60,000 comparisons we can make of statistical tests. And then we compared the performance of the 2 libraries with respect to quantitation. And the benchmark criteria we used first was very simple, just proteome coverage and missing values. That's something you can look at relatively easily. Then precision of quantitation, so we expressed this using CVs, and also accuracy. Since we knew how much we spiked in from the UPS2 standard, we can easily, or relatively easily, also look at accuracy of quantitation. And in the end, what really counts for us the most is the quality of the candidate list in terms of differential regulation, because what we mostly are interested in the end is how good does the candidate list reflect what you are biologically interested in the end. And this is also what we normally, or this is the single most important thing our customers want to know from us when we perform an experiment for them. So that's why this has really a high weight for us. So the first results, the comparison in terms of the identifications and the missing values shown here. As you would basically expect, you get a little bit better coverage with the project-specific library, not much. However, very interestingly, in terms of the UPS2 proteins, the hybrid library performed better. So that was very interesting. If you look at the missing values, again, actually what we would have expected, the hybrid library performs a bit better in terms of reproducibility of quantitation. The difference is not orders of magnitude, but it's a little bit better. Also, of course, you always have the effect, once you go deeper in terms of coverage, your quantitation will get more sparse and also of lower quality. That's obvious, right? It's always more usually the case. Yeah, but what we are really interested in is why is the UPS2 proteins better reflected in the hybrid library analysis as compared to this project-specific library. And I think this can be relatively easily explained, the way how we generate this project-specific library. So this is a hypothetical example where you would have 10 samples, I think it is, 9 samples. And you have a protein of interest, in this case could be a UPS2 protein, and it's only high abundant in a few samples. Now, in the standard way of how we build the libraries, we make a pool of all those samples and we build the library based on this. But because the signal of this protein here is really diluted in the pool of samples, it's not so easy to pick it up while you build your library. However, when you are doing a direct DIA way of analyzing your data, you will search each and every run of your samples separately. And in the runs where your protein is really highly expressed, you have a higher chance of picking it up also, because here, relatively speaking, it has a higher intensity. So, and that's actually an important effect, because you are mostly interested in the proteins that differ in the expression level between your runs, and not the proteins that always have the same expression level. So that's quite quite important. Then we looked at the next benchmark criteria, the precision and the accuracy in terms of quantification. Here you can see how we calculated this, coefficients of variation of the background proteins. So here it's on protein level, that's important. What is included here is the biological variance. Remember, these were distinct mice. So within this variability, the biological variance is included. When you look at this, you can see that the hybrid library performs a bit better than the project-specific library, which is, yeah, you could say is maybe a bit expected because of the reproducibility of quantitation. In terms of the accuracy, there's not so much difference. It's actually flipped over. And yeah, what is really important here, so first of all, I would say the differences are relatively small, but much more important, when you are performing such kind of a comparison, always bear in mind that this is confounded with the protein coverage you have. So in one way we had a slightly deeper protein coverage, in the other we had a slightly lower protein coverage. So now if you actually do this on only the overlapping proteins between the two analyses, it looks more like what you would expect. Basically these two benchmark criterion look the same. So in terms of precision and in terms of accuracy, they perform virtually the same. And this is actually also what we would expect, because if you identify something, the quantifications should be similarly good between the two, right? So maybe it makes a difference what you can identify, but once you did identify and your FDR estimation is correct, the quantitation should be the same. And this is what we can see when we do this comparison. Now to the most important benchmark for us. You, again, as I said before, you can make these candidate lists where you would have compared two conditions. You can rank this by p-value, and you can check how many of these candidates correspond to my UPS2 proteins in this controlled condition. And we did that and compared between the two libraries, and this was the result we got. And it's actually quite a striking result. So in terms of performance for the candidate list, there was quite a significant difference between the hybrid library and the project-specific library. And it was not in a direction that the deeper coverage automatically led to a better candidate list, because we could identify more of the UPS2 proteins. So this really made the big difference in this way of looking at the data. Maybe also interesting to note, this line here would correspond to the perfect candidate list, meaning if you look at this point here, I would have a candidate list of the top 50 proteins in my candidate list, and all of those would be UPS2 proteins. So all of those would correspond to proteins that you knew they were differentially abundant, and there were none of the background proteins in there. So it's really up to this point almost a perfect analysis. So yeah, this was really also quite striking to us, that the hybrid library performed quite a bit better than what our normal way of analyzing the data. Now finally I also have, I still have plenty of time, I also have like a showcase example. It was not a customer project we did, it was more a project that we did ourselves just to test the different kinds of workflows. And here you can see the setup. So we had 24 samples from 12 individuals, 12 times healthy tissue. So these people, they had cancer and the tumor was excised. And while taking out the tumor, also some of the healthy tissue was cut out. And then this means you always had parts healthy tissue and parts tumor tissue from the same patient. 12 times healthy, 12 times cancer tissue. And we also have two different cancer subtypes in here as determined by a histological analysis of the tissue, which was adenocarcinoma and squamous cell carcinoma. Then we did our standard way of acquiring the data. Here you can see the details. In total, that's less than 3 days of acquisition. for the 24 samples. We actually did this in Bremen back then because this was done before the launch of the HFX. And we analyzed the data then with Spectrum Pulsar X. Yeah, not much mention here. We, like what we normally do to avoid any bias in the label-free quantitation, we did a block randomized acquisition of the data. And we built a hybrid library as well. Actually, I'm not showing this, but also a project-specific library for this data set. And the hybrid library we generated again with this very big data set here, which in our hand really performs well for everything like cell lines or human tissue. It's a really good resource to do that, at least in our lab. We performed direct DIA. In this case, because you have quite a bit of variability among the humans and among the cancer tissue, even this gets pretty extensive, pooled together. And the hybrid library was this size in the end. Applied to the data, it looked as shown here. So this is now a bit more detailed comparison. On the left, the project-specific library. This we did really quite extensively. I think it was 30 runs, 30 fractions. Then the resource library and then the hybrid library. And we show this for cumulative protein identifications, so over the complete experiment, always with a 1% protein FDR. Then in terms of average IDs and in terms of complete IDs. And quite striking in this example here, the hybrid library always performed best. It's not a huge difference, but especially in terms of the consistency compared to the resource data, it's quite a big difference. This is then when you look at this data from a bird eye perspective, you could say. We have done this also using the project-specific library. It looks very similar. Also, when you use the resource data library, it looks very similar. But what is nice to see is if you perform an unsupervised clustering on this data, you can very, very easily distinguish healthy tissue from cancerous tissue here. So this is actually a Z-score transformed protein matrix, what I have shown here. So it's profiles of protein quantities. And there is a huge body of proteins that are downregulated in healthy tissue and a huge body of proteins that is upregulated in healthy tissue as compared to the cancer tissue. And then what is also interesting is that in terms of cancer subtypes, we also almost get a complete separation of these two cancer subtypes, which was also quite striking to us to see. The same we could actually see when we did a PCA analysis of this data. Also there we could see that healthy and cancer tissue perfectly separated and the two cancer subtypes almost perfectly separated. Yeah, also in terms of data completeness on protein level, it's quite complete. So it's really nice. And then we did a little bit more in-depth analysis. Again, here I did this analysis before using the project-specific library. But these data here, they look essentially the same. So it's a really high quality results you get. We can also cross-compare this to published data and we get exactly the same. It's actually shown on this slide here. So we compared it to the paper of Stefan Tänzer. It was actually a very similar setup, interestingly. And we could confirm 5 out of 8 proteins, they reported as significant in our data set. Doesn't mean that the other 3 were not correct, but this was a very stringent filtering on our side, and 5 of the 8, they also passed through this very stringent filtering. So meaning same direction of regulation and a very low Q value. And interesting was that we could really separate cancer subtypes from each other in terms of proteins that are differentially regulated. And that's, of course, the more interesting part, because if you're thinking along the lines of personalized treatment of these cancer patients, you would like to read as much information out in terms of subclasses of cancers from these patients to be able to make a good prediction which medication would work best for each of the patients. Yeah, I'm a little bit too early. It's already the end of my talk. One thing I wanted to say here, so all these has been done without any overhead in library generation. That's quite remarkable. So we didn't acquire any extra DDA runs to analyze these DIA data. So that's really, for us, quite a big step forward. And that's also why we want to use this now more in our laboratory to really reduce the LCMS time needed for the analysis. So in terms of the conclusions, building hybrid libraries from resource data and directly analyzing the DIA data comes with some challenges, and these have been basically addressed in the latest release of Spectronaut. One of those challenges is the dilution of the iRT precision, which has an impact on the reproducibility of quantitation, as I have shown. And then this tiered iRT approach can solve this issue. And then this, as I said before, right, quite striking, these optimized hybrid libraries, they really perform a similar or even better performance than the project-specific libraries. And I had some explanation it was some hypothesis that will have to be tested, but we think we have an idea why that's the case, and it's really quite striking. And finally, it renders, so using these hybrid libraries, basically renders the project-specific libraries unnecessary, which is actually great, because this has always been something that makes the workflow a little bit more complicated and tedious and lengthy as well. Okay, thank you very much.
Great.
Yeah, of course, I would like to thank my team in Zurich here, and then also our collaborators at Thermo and Ting and Olga. They actually worked on these quantitative controlled data sets together with us.
Great, thanks, Lukas. Any questions on that? Yeah.
Have you compared your hybrid approach with the SWATH extend, they call it the extended library?
No, we did not, no. I think I've heard about it, but we did not compare it, no.
Would you have an idea on how Spectronaut performs when it comes to PTMs, and especially to centralization, confidence, or whatever extra step you would have?
Mm-hmm. Yeah, I mean, it was not really a topic of my talk, but in Spectronaut there is no specific scoring for PTMs implemented at the moment. It's something we want to work on actually. But there are certain things you can do to improve the analysis with PTMs. In every DIA software, I guess it's the same. What you can do is, for instance, when you do phosphoproteomics, which we have also done in the past, you can include more of the fragment ions into your library being used. So in Spectronaut, for instance, there's a setting to do that. In other software, I guess there is the same thing, just how stringent do you filter how many of the fragment ions should be used in the analysis afterwards. And of course, the more fragment ions you are using, the more likely you are that the scoring of Spectronaut will realize that there is a difference between the different forms of the phosphopeptide, right? So you can bias it more to the right way of analyzing. And then the second thing you can do is what I have shown here, actually, this precision iRT. So as soon as you have very, very precise iRT and you really know where to look very, very accurately, you can also more bias yourself to the right peptide with the right PTM. Of course, often these peptides elute very, very similarly, and then it will not be possible, right? So that's another thing you can do. And then the third thing you can do, if it's about precursors that have distinct precursor M over sets, some of them don't, right? If you have a phosphor on the same peptide that hops around, you do not have that. But if they have different precursor M sets, you can also make methods with more windows, faster methods. And then you have less of these precursors that fall within the same windows. And that's basically what we have done when we did phosphoanalysis. And you can do phosphoanalysis and it works, but you have to go through the results manually through the interesting candidates and check whether everything is kosher if you want to be on the safe side, and it can certainly be improved, that's for sure.
One more?
If you're fractionating and using different DDA runs to build libraries, how do you merge the different fractions together?
Can you say that again? If we fractionate to build the libraries,
how do you merge the different DDA runs and the different fractions together?
Which information are you talking about? The retention. We just simply take the median of the iRT, determining each run separately.
But they have not much overlap, presumably.
Sometimes you do. Normally, if you do fractionate, the less the less overlap, the better, right? So ideally, you would want to have every peptide in only a single fraction, but sometimes you do not have this case, and then you have maybe 2 or 3 values. Then we just simply take the median. But if this analysis works in every single run, and if you do not have very heterogeneous data, then the median will not make a big difference to whether you just take a single of these values.
So you don't try and align like gradient differences between the different runs?
No, we don't have to do that because we do this local regression in every single run. So the normalization is happening already at the run level. And once we have normalized on a single run level, we already have iRT, that's a normalized value, and that's a comparable value. So we do not have to do any more alignment between the runs.
Are those MS2 based iRTs, or you have some feature? You have a MS1 feature based way to do this?
You mean whether it's based on the MS1 or MS2 level of information?
Yeah,
it always
scans?
depends on both levels of information. So we determine peak boundaries not only based on MS2 information or only based on MS1 information.
I rather meant the library though. So when you're deriving the library IRTs, so we've always done this based on just take the median or something like this of the MS2 scans that where this peptide sequence was identified. But you could imagine, now that you're getting down to these very fine tolerances, you could imagine having a MS1 feature-based retention time that might be more accurate. Is that something you thought about? Or?
Yeah, I think I know what you mean. So I can just tell you how we do it. We never tried anything else. So we just basically determine peak boundaries over retention time dimension. And then you can also determine the apex between those boundaries.
But that's in the MS1, right?
And that's based on both levels. So the boundary determination is based on the MS1 and MS2 level.
But in the if you're building a library generation from DDA data, then you don't have any peak reconstruction from MS2 data, right?
Sorry, yeah, that's done on DIA level.
Okay.
On DDA level, it's just MS1.
Just MS1. Okay.
Yeah.